Hey, are there any questions?
No.
All right. Well, let's give the students another minute to log in. There's only a few people logged in. And then we'll begin. Let's see. I need to open up chapter 7 anyways. Uh, okay, can you see my screen?
Yeah.
Okay, thank you. This is the last slide we were talking about. How heat is not as efficient at transferring, I guess, heat compared to wet heat. I know that didn't quite sound right, but uh, wet heat is much more efficient in transferring heat, and that's why you can't put your hand over steam, but you can put your hands in a hot oven. Let me go back to the syllabus and say that we're going to finish up chapter 7 today. And if I have time at the end of this lecture, I'm going to give a review for the final. I'm hoping to get the review finished in the lecture, but if I don't finish in the lecture, we'll come back in the lab and finish the review. Remember that the cumulative and comprehensive final is due by 11:59 p.m. on Wednesday, March 17th. That is next Wednesday. Your unknown project and the unknown notes are also due this week, and I think both of them are due Saturday at 11:59 p.m. But let me briefly check on that. I know that's when the unknown lab report is due because that's on the syllabus, but I think that's when the unknown notes is also due. But let me check on that. You will be pleased to know that I have taken a class on becoming a better online instructor, and hopefully I passed that class. The class just finished. Was it this week? I haven't actually gotten my grade yet for it, but I think it finished. All right, there's the unknown notes page. It says March 12th. So no, the unknown notes page is due before Saturday. You want to finish the unknown notes page on Friday. Any questions about that? All right, let's move on. I can get it. Here we go. So another physical method for controlling microbial growth is filtration. You have a liquid or a gaseous solution, and then you pass it through the filter, as they're doing here. And the filter has small pores in it, allowing the liquid or the gas to move through it, but preventing the microbial growth from getting through it. Filtration is most often used with liquids for sterilizing the liquid, meaning that the liquid down here is sterile. Some examples for using this are for juices and Coors beer, vaccines, which uh, filters it and then doesn't change the taste, whereas usually if you heat up the juice or the beer, like pasteurization, it changes the flavor. And that's why some people really like Coors beer, because it has the unpasteurized taste. And I don't know if you guys are beer connoisseurs or not. My brother used to go out of the state and buy Coors beer, because in the state of Oregon, they made a law like in the 1920s, all beer must be pasteurized. And Coors wasn't allowed to be sold for a long time. I think they finally got it changed in 1980. And the pasteurization law was just to make the beer safer so that people wouldn't consume food poisoning when they consumed the beer. And Coors filters their beer, which solved the problem. And I think it was in the 1980s the state of Oregon allowed Coors beer to be sold. Filtration usually isn't used for milk because the proteins and the fat in the milk clog the filter. So it's only used for juices and beer and, like I said, some vaccines. Any questions about filtration? Low temperature is a physical method for controlling microbial growth, but it doesn't really kill the microorganisms. It may inhibit their growth if you bring the temperature too cold, like a hypothermophile will not grow in the refrigerator. I think most mesophiles do not grow in the refrigerator. The trouble is you bring the food out and you serve it, the food warms up, the mesophiles can grow, and then you put it back in the refrigerator, it doesn't kill them, and then you bring it out again. So mesophiles can cause spoilage in refrigeration, especially when they're growing before the food gets cold in the refrigerator. But if it is strictly refrigerated in refrigeration temperature, generally the mesophiles are inhibited in their growth, and the other microorganisms that grow in the refrigerator are the ones that cause food spoilage. And they can cause food spoilage within a week, and that's why for most foods, when you have it in the refrigerator, you should throw it out after about a week. It just slows the metabolic rate of the bacteria. It does not usually kill them. It does not destroy any toxins. Yes, I already talked about that. Freezing also does not kill 50% or more of bacteria. It does kill some bacteria because water, ice crystals form and it destroys the cell. But bacteria are fairly resistant to freezing. And if you had a population of them and then you froze the whole population, only about 50% of the cells would die. 50% would be alive. So freezing also does not kill microbial growth, at least not very efficiently. It inhibits their growth. And freezing actually stops the metabolic activity. Like if you put something in your slow freezer, your home freezer, that freezing does kill all parasitic eukaryotic species. The ice crystals form in them, and eukaryotic cells are larger, and the ice crystals will kill the eukaryotic cells. This is slow freezing. If you were to put things in liquid nitrogen, you essentially would not kill any of the bacteria cells. All right, any question about that? All right, didn't hear any questions, so let's move on. High pressure is another physical method for microbial growth. High pressure transfers pressure evenly throughout a liquid, killing vegetative bacterial cells. It does not kill endospores, but it does kill vegetative cells. It denatures proteins and alters carbohydrates. Endospores can survive the high pressure, but we use high pressure to preserve the color, the flavor, and the nutrient value of juices. So oftentimes in the store you will have a juice and it doesn't taste like it's been pasteurized because it's been pressurized, and it kills the endospore, not the endospore, the vegetative bacterial cells in it. And here's a little joke about pressurization. The mother's talking to Junior and says, "Your dad's been under a lot of pressure lately." And as you can see, they're charcoal, coal, and father comes home as a diamond. So endospores are resistant to high pressure. They can survive. I'm not sure, because they sell the juices and they would have endospores in them when they've been pressurized. And I guess the endospore numbers are low enough that it's not a health concern. Desiccation and osmotic pressure are essentially the same method, where physical method to control microbial growth. Desiccation is drying. Osmotic pressure is having something like high salt or high sugar around, and then the water leaves the bacteria. Desiccation prevents microbial growth, like when we eat dried fruit. The bacteria cells can remain viable for years if they are endospores, but they cannot grow and reproduce. Some examples, I mentioned dried fruit and figs, and some examples of osmotic pressure, molasses and honey. You can keep those things around for years. My mother had a bottle of molasses, which was, we figured, 20 years old, and I made molasses candy for my father, and he ate it and enjoyed it and didn't suffer any ill effects from it. So molasses, high osmotic pressure kept that from spoiling. And the bottle was, we figured, 30 years old, hadn't been used for 10 years, and was still good. And we decided, well, we're not using it, so we finally threw it away after about 30 years. In high osmotic pressure, what happens is the water leaves the cell, the plasma membrane pulls away from the cell wall, and you can actually see it. If you happen to, well, you guys wouldn't have seen it in biology. What's the biology prerequisite for this class? Is it 160?
Yeah, it was 160.
Yeah, 160. You guys probably didn't get to see that because you're doing that online, but you can take a look at putting a cell, usually an algae cell, in high osmotic pressure, and the plasma membrane pulls away from the cell wall, and you can actually see it under the microscope. And that's from high osmotic pressure. That tends to kill most bacteria cells. Endospores would survive high osmotic pressure and desiccation, but the drying out tends to kill most vegetative bacteria cells. Another physical method for controlling microbial growth is radiation. Radiation damages DNA. Ionizing radiation is radiation that we call things like X-rays, gamma rays, and electron beams. They have a short wavelength. Here we're looking at all of light. There's visible light. And usually when we're talking about radiation, which is actually all of this spectrum, the electromagnetic spectrum, but when we talk about radiation, we mean something from UV light down, what is that, lower or higher? Shorter wavelengths. Ionizing radiation ionizes molecules and water. Of course, water is a molecule. That hits the electrons and gets them to leave the molecule, creating the ion. And it can also create hydroxyl radicals. And these molecules react with other organic molecules, especially DNA, and that damages the cells. You get a bacteria cell gets hit by ionizing radiation just a couple of times and you mutated the cell. And then after a few mutations, you killed the cell. Ionizing radiation is used for making some food safe. You can buy it in the grocery stores. It's the food that's perishable on a shelf, and it'll be wrapped and ionized and it doesn't spoil. Some pharmaceuticals are sterilized that way, and then some medical supplies. Non-ionizing radiation in your textbook author means ultraviolet light when they use the term non-ionizing radiation, results in a bond formation between adjacent thymines. So two thymines that are next to each other, when they're hit with UV light, instead of binding with a hydrogen bond from the T to the A, they actually form two covalent bonds between two thymines. And that puts a bubble in the DNA. And this region of the DNA cannot be synthesized in DNA replication, and it cannot be transcribed. So that's how UV light kills cells. UV light is good at killing cells, but it doesn't penetrate, so UV light is usually only used to sterilize air or surfaces of an object. If you have a flat surface, it's easy to sterilize. But if you had a cube, you'd have to go in there and rotate it so that every side would be exposed to the UV light. Microwave radiation is not particularly antimicrobial. Microwave radiation does kill cells by creating heat. When a microwave hits water, the water molecule absorbs it and then becomes hot, and then that heat can kill cells. So that's how microwave radiation kills cells. Not directly, but the microwaves are transformed into heat, which is another form of energy. All right, this table is giving you a summary of the different physical methods for microbial control and then comparing them. You don't really have to memorize this table, but you probably should know the different methods. You don't really need to know the mechanism of action. You might know a little bit about the comments, but you don't have to memorize it. And then preferred use is just really for your own personal use. And then a few other methods for controlling microbial growth. Let's talk about them. Moist heat, very efficient. Autoclaving, probably the most efficient. Pasteurization, that's also moist heat. Dry heat, not as efficient. Direct flaming is efficient because of the very high temperature. Incineration and direct flaming. Hot air sterilization, and you'll note in here it takes about 2 hours to sterilize things. Whereas pasteurization, which doesn't really sterilize, but autoclaving, it takes only 15 minutes. Filtration, also effective. Cold, this only generally stops growth. It doesn't sterilize. High pressure, it does kill vegetative cells, not the endospores. Desiccation, similar to high pressure. Osmotic pressure, similar to desiccation. Radiation, very good at, especially ionizing radiation, very good at killing off microbes. Non-ionizing radiation, it takes a little longer, but it's also good if you can expose for, I don't know, 15 minutes kills all microbes. All right, enough for talking about physical methods for controlling microbial growth. Let's talk about chemical methods of controlling microbial growth. And let me state that there are few agents that achieve sterility on all microbes. Most microbes one agent does not kill everything. It does decrease their microbial numbers to safe levels, like when you disinfect the toilet, you don't sterilize the toilet. Is that clear? There are some microbes that survive disinfecting the toilet, but it does bring the levels of microbes down to safe levels. Few disinfectants will kill endospores, but they are pretty good at killing vegetative cells and then pathogens. And even with them, disinfection usually does not sterilize the object. It only brings the microbes down to safe levels. So disinfection is termed the destruction of vegetative pathogens. The endospores are not killed. There are various factors related to how effective the disinfection will be. The concentration of the disinfectant determines how efficient the disinfection will be. Most agents, the higher the concentration, like with bleach, the better the disinfectant. But that isn't true with alcohol. With alcohol, you want the concentration to be around 70% alcohol, and that will be the most efficient at killing microbes. If you go above 70%, like 95% or 100%, the alcohol is less efficient than killing the microbes. Any question about that? Time is also a factor related to disinfection. Generally, the longer the time, the better the disinfection. So the longer the disinfectant is applied, the better the killing will be. And that also goes along with, you have to have access to the surface to be disinfected. As I mentioned with UV light, it's very good at killing off the surface, but you have to have that surface exposed. The underside is not going to get any exposure to UV light. pH can be used to disinfect high acidity or high basicity, I don't know what you call that, basicity, will kill vegetative cells. It depends on the species which pH is good at killing the cells. For example, the bacteria that live in your stomach would survive fairly strong acids. But normal pH and higher pHs, meaning something that's basic, would kill off those cells. The presence of organic matter, which is often found in clinical situations, can affect the disinfection. That many disinfective agents are inactivated by organic matter. And that would be like fecal samples or vomit samples, or even dandruff has organic matter, and that prevents the disinfection from working. Something that's very inactivated by organic matter is bleach. Bleach is a very good disinfective agent, but it is inactivated by organic matter. So if this is missing, then the disinfectant works generally pretty well. And if it is present, that means you need to use more of the disinfective agent. All right, this table is listing the different types of disinfectants, and let's look at the next table. The table is...
What would iodine be considered then?
Is somebody asking a question?
Yeah, I was. Sorry, I don't know if you can hear me or not. What would like iodine or like betadine be considered then?
Iodine is a halogen. So is bleach.
Oh, gotcha. Okay.
Okay? So heavy metals and their compounds, let me blow this up because that's not very visible. Let's put that. Bottom, there we go. Am I missing the first one? Yep. Heavy metals and their compounds, denature enzymes, and essential proteins. Silver nitrate used to be used on children all the time to prevent the risk of them coming down with gonorrhea from birth. But now these heavy metals have come back, and they're using them in Band-Aids or dressing, where you can buy a cream. And it's just that they tend to prevent microbial growth. And I think they're coming back because a lot of bacteria are becoming resistant to antibiotics. And so we're using more of the heavy metals than we did, say, like 20 years ago. I don't remember when they were used, like silver nitrate. The doctor always used to treat the baby with silver nitrate after he was born. I think they stopped doing that in the 1970s or even earlier, but that was something that happened very frequently in births in this country. I don't think it's continuing today. Surface active agents, we're talking about soaps and detergents, not acid anionic sanitizers. They tend to remove the microbes through scrubbing and washing them away. They're not very effective at killing microbes, but they are effective at washing them away. So you use the soap and the water, and then you reduce the number of microbes like on your hands when you're washing your hands. You can put antibacterial agents in the soaps, and then they are more lethal. And then the hospitals use quaternary ammonium compounds. These are more efficient at killing microbes. They are an antiseptic for the skin, instruments, utensils, and rubber goods. Some examples, if you ever worked in the clinic, you may have heard of Zephyrin and Cepacol. Quats, they're also called quats. Let me see if I can move that just a little bit. Didn't mean to do that. I gotta get this out of there. There are some preservatives that are put in food, organic acids, nitrates and nitrites. They prevent the growth of certain microbes, and they're just added to food to help the food keep better. A lot of meats have nitrites and nitrates in them. It prevents the growth of Clostridium botulinum and also helps protect the food from spoiling. And the organic acids, let me see if I've got a name here. It's mostly to prevent the food from spoiling, preventing mold mainly, but other microbes too. I'm trying to remember some foods that have that in them. Obviously meats and hot dogs have nitrites and nitrates. bacon, another thing. Doesn't say the food. Bread. There it is. Calcium propionate is used in bread to help preserve the bread, keeping the mold from growing in it. If you bake your own bread and you don't add any preservatives to it, which most of us don't, after about a week at room temperature, it'll become moldy, assuming the bread is moist. But if you notice, when you buy store-bought bread, they can last for about a month, and it's because of the organic acids that they add to it. Aldehydes, they cause protein denaturation, very effective antimicrobial, glutaraldehyde, formaldehyde. That's usually used for disinfecting medical equipment because aldehydes are somewhat toxic to people. Ethylene oxide, a very good sterilizing agent. It sterilizes essentially everything, although it takes a long time. It does kill endospores. It has hydrogen peroxide in it, and that's probably why it's killing endospores, and that is, endospores are anaerobes, and you bring in reactive oxygen molecules like ethylene oxide and hydrogen peroxide, and they start killing the anaerobes. We'll talk a little bit more about how we can kill endospores, but this would be one way. The trouble with ethylene oxide is it's highly toxic to people, and you need to expose it for a fairly long time, like something like 20 minutes, to make sure you kill all the endospores. Plasma sterilization. We use this for tubular medical equipment, usually using hydrogen peroxide, which we'll talk a little bit about that later. Oh, there it is. I'm not sure why that's there if it's using hydrogen peroxide. It should be down here. And then supercritical fluids. They use this for sterilizing organic medical implants. And I'm not sure how this works, but carbon dioxide compressed to a supercritical state is used to help sterilize fluids. Peroxygen and other forms of oxygen kill cells by oxidation. Hydrogen peroxide is the most common household agent, which is peroxygen, and it is very good at killing endospores and all microbial growth. So if you want to sterilize something and you want to remove the endospores, get some hydrogen peroxide and use that as an antiseptic. Realize that if you put it on your skin, you must use copious amounts of it, because our own skin produces catalase, and catalase will inactivate hydrogen peroxide. But if you use copious amounts of it, you will kill the bacteria on your skin. So it is a good disinfectant, and it is toxic to our cells, but it can't get through our skin. You can take diluted hydrogen peroxide. I think you dilute it 1 to 3 and then use it as a mouthwash. I'm not sure why you're supposed to dilute it. It does kill some of the cells in your mouth, but not very many, especially if you use 1/3 dilution. And it is a effective disinfectant. We'll just word it as an effective disinfectant, and one of the few that kills endospores. All right, any question about that table? No, let's end the show and talk that you can compare the effectiveness of various antiseptics, the percentage of bacteria surviving, and then the time of exposure. And as you can see, soap and water, even after, 2 minutes, you don't kill most of the cells. You only kill about 10% of the cells. and nobody washes their hands for 2 minutes. Okay? On the other hand, if you look at 1% iodine tincture of iodine, after exposure to a little over a minute, maybe a minute and a half, most of the cells are killed. Now realize this graph stops at 10%, so to get 100% killing, which usually even in the clinic they aren't concerned with 100% clinic killing, you'd have to have long-term exposure, even of that one. But the point is, when you apply it to a patient's skin, and then it just sits there, after about 2 minutes, the vast majority of the microbes are killed. All right, any question about any of that? When we compare the different ways of killing microbial growth, it's a simple hierarchy, and this is a little bit oversimplified, but we can show the relative resistance of microbial groups to chemical biocides in general. Prions are the most resistant microbe. Remember, these are just infective proteins. They're very, very difficult to kill. You have to take them up to 1,000 degrees Fahrenheit to kill them. And most other disinfectant agents, even radiation, are pretty lousy at killing them. I think with radiation, you need something like several hours worth of ionizing radiation to start damaging the prions. So even killing them with radiation is very difficult. Endospores are the most resistant living microbe to disinfectants. They're very difficult to kill. There are a few agents, though, that are pretty good at killing them. Like I said, hydrogen peroxide and the other peroxides. Ethylene oxide gas can kill them. Most endospores are resistant to most other disinfectants. There are some species of endospores which can be killed by bleach, but most species of endospores are not killed by bleach. And it just depends on the species and how they form their endospores. Mycobacteria is the next most resistant thing. Most disinfectants or antiseptic agents are soluble in water, and so they have trouble getting through the mycolic acid in the mycobacteria. Okay? Let me state on this one, there is a disinfectant, which is a lousy disinfectant, which is actually very good at killing mycobacteria, and that is alcohol. Alcohol goes right through the cell wall of the mycobacteria and can kill the mycobacteria. Alcohol is a lousy disinfectant. As we mentioned, we don't even use it as a disinfectant. What we do is we use it as a degerming agent. I'm trying to show you my skin. Like when we apply it to the body before we give an injection. We're using it as a disinfectant agent. Is there going to be a question? All right, let me move Denise. She was making noise there. The cyst of protozoa is the next most resistant. You have to have specific treatments to kill the cysts of certain protozoa, and that's why they survive the stomach. When you consume a cyst of a protozoa, and it can survive the stomach acids, they're pretty hardy. Vegetative protozoa, the next most hardy thing to most disinfectants. Gram-negative bacteria are a little bit more resistant to gram-positive bacteria because of their outer membrane to their cell wall. fungi, including most fungal spores. Viruses without envelopes are a little bit more resistant than most disinfectants than virus with lipid membranes. Then we have gram-positive bacteria. And then the easiest thing to kill are viruses with lipid envelopes. That lipid envelope is easy to disrupt, and that inactivates the virus. If you remember, coronavirus is a lipid membrane bound or enveloped virus, so it is a virus that is easy to kill. Any questions about all of that? So this is a general hierarchy. Like I said, there are some exceptions, like alcohol is generally, it wouldn't kill endospores at all. It would be very poor at killing cysts. It would not be very good at killing gram-negative bacteria, but if you give enough exposure time, it would kill the gram-negative bacteria. And my point is, alcohol isn't very good as a disinfective agent, but it is fairly good at killing mycobacteria, which is resistant to most other disinfectants because they are soluble in water and don't get through the mycobacteria cell wall. And I said this is a general thing that we're talking about. In principle, the gram-positive bacteria are more sensitive than the gram-negative bacteria, and it's because of the outer membrane. The disinfectant has to get through the porins in the outer membrane of the gram-negative before it can affect the gram-negative cell. And I've already mentioned endospores. Very few disinfectants kill endospores. There are a few exceptions, mostly the peroxides. Already talked about viruses. And then I talked about prions. How do we kill prions? The solution has not exactly been solved yet. With Britain, when they had the mad cow disease, what they did was they incinerated the carcasses, heated them to 1,000 degrees Fahrenheit to destroy the prion. With surgical instruments, you cannot do that. Expose them to 1,000 degrees Fahrenheit, because most surgical instruments will be damaged, at least with prolonged exposure and repeated exposure to that temperature, because that's so high. But they found that there is one agent that can treat surgical instruments, and that is you treat them to proteinases, an enzyme that degrades proteins, destroys prions, and they do use that on surgical instruments to prevent prions. So if ever they're working on a patient and they wonder if the patient has prions, they would treat those surgical instruments with proteinases. And I do not know if in the hospital they routinely do that, or if they only do it when they suspect there could be prion infection in the patient. All right, while microbial control methods are effective, especially the biocides, they are not uniformly effective against all microbes. The compounds discussed in this lecture are not generally useful in disease treatment, except for the antibiotics. Antibiotics are used in people to treat diseases, but most of the other are not used in treatment, and the reason is because they are toxic to people. If you remember, our former president said, gosh, bleach is effective at controlling coronavirus. What we should do is have people take bleach. Well, bleach is toxic to people, and most of the other disinfectants are toxic to people, and so we cannot use them for treating a patient with an infection. You can use, however, antibiotics to treat the patient because they are not toxic to people. Any questions about any of that? Okay, if there's no questions on this lesson, I'm going to wrap it up here and begin the review. I don't think we'll finish the review, but I'll get started on it. What I'm going to do is just go through each chapter. And if there's any questions in this review, please ask. Chapter 1 and a little bit about Chapter 10. You should know the history of microbiology. Who discovered what and what they discovered. Okay? Like we ran into these topics time and time again, so you should know this. We got an introduction in the first chapter, but we talked about Koch numerous times, Louis Pasteur numerous times in the lesson, and I like history, so you should study this, okay? Any questions there? I just gave you a hint. I like history, so you're going to be having a few questions on the history of microbiology. Let's see. Well, you should know a little bit about the importance of microbes. And then these terms, we ran into them in various places, like pasteurization. We talked about that in 2 or 3 places besides this lesson. And then Koch's postulates, we ran into it 3 times in the term. And so you can imagine there's going to be a question on Koch's postulates on your final, because you ran into it 3 times in the class. All right, any questions about Chapter 1? If not, let's go on to Chapter 2. Having a hard time finding the PowerPoints. You should, with respect to the 4 biological molecules, be able to describe the relationships and the characteristics of monomers and polymers, and describe the composition and structure and function of the 4 major biological molecules. Meaning, if I show you a picture of a major biological molecule, you could tell me which of the 4 groups it belongs to. Okay? So give an example of each. Any question about Chapter 2? Chapter 3, observing microbes. Well, you should understand the sizes of organisms and the properties with relationship to the properties of the microscope. And I'm not going to ask you to know the exact size of an organism, but you should be able to tell me that most viruses are smaller than prokaryotes, which are smaller than eukaryotes. So you should give me the relative order. You should know the properties of the microscopes. And you should know the 3 types of stains: simple stains, differential stains, and special stains. We discussed this, I think, in 3 places, in 2 different labs and in this lesson. And then, of course, you should know the Gram stain, which is a differential stain. You should know its procedure and the basis of its differential staining. Any questions about Chapter 3? You probably should know about peptidoglycan. We talked about that, I think, 3 times, at least twice. And we went into its construction in detail. I don't remember this lesson or the next lesson, but one of them. On this lesson, we also had the metric system, and I believe there is one question using the metric system, and you do need to know how to convert in the various metric units. Chapter 4. You should know the different functional groupings and structures of prokaryotic cells. I'm unlikely to ask you anything about eukaryotic cells because you were supposed to have had that in Biology 160. You should also know, like I said, I think I kind of talked about this, the cell wall and peptidoglycan, to know the differences between the gram-positive and the gram-negative cell and their cell walls. And we did run into that. I think we ran into that at least 3 times. Twice in the lecture and then once in the lab. At least 3 times, maybe more than that. Is there something else on there? No. All right, any questions about Chapter 4? We're going quickly through these early chapters because you've already been quizzed on them. We will slow down when we get to the chapters after the quizzes, and that will be chapters after Chapter 8, with the exception of Chapter 7, which we just did. Chapter 5, controlling microbial metabolism. Well, you should know the trophs, like the photoautotrophs, the photoheterotrophs, the chemoautotrophs, the chemoheterotrophs. And if I give you a bacteria, tell me which troph it belongs to. You should know about fermentation and aerobic and anaerobic respiration. We spent a long time talking about aerobic respiration in this lesson. I won't ask you about non-cyclic photosynthesis. I might ask you about cyclic photosynthesis. I don't remember if I've got a question on the quiz or on the final on cyclic photosynthesis. So it's best to study that one, and then aerobic respiration and how it differs from anaerobic respiration, and briefly understand fermentation. That's actually a simple process. And the trophs. So that's a fair amount in this chapter. All right, any question about metabolism? Chapter 6. Well, you should know the different terms used for describing microbial growth, like the psychrophiles, the psychotroph, the mesophiles, the thermophiles, the hypothermophiles. These are all temperature trophs. The acetophiles, they live in acid environments. Halophiles, they live in high salt concentrations. You should know about growing microbes in thioglycolate broth, and the thioglycolate broth creates an oxygen gradient in the tube, and you will get the anaerobes growing in the bottom of the tube, the aerobes growing in the top of the tube. The facultative anaerobes will grow everywhere, but they'll grow best where there's oxygen, so they'll have their most growth at the top of the tube. The aerotolerant anaerobes will grow everywhere uniformly in the tube. And then the microaerophiles will grow best midway, someplace in the middle of the tube. You should know about how microbes grow. Most of them grow by binary fission, but there are some that reproduce by reproductive spores or by budding. You should know about endospores. We actually discussed that earlier, but in this lesson I'm going to tell you that this is not cellular growth. It's a cellular survival species, so you should know the difference between an endospore and a reproductive spore. You should know the various requirements for growth and that there are 4 phases of growth. And I think I have an essay question, but that could have been on the quiz. I'm sorry, I'm getting my quizzes mixed up with the final. But you should know the 4 phases of growth. All right, any question about chapter 6? Let's go on to chapter 8, and we'll come back to chapter 7. Well, you should know the terms of chapter 8. You should know about operons and how they work. That is describing gene expression. Operons are involved in gene expression. You should know the 3 ways that genetic material can move around from cell to cell in the same generation of cells. And where do I have that here? It doesn't look like I've got that here. I should have that up here probably. 3 methods of horizontal gene transfer. Drawing a blank here. Transduction. There's 1. Conjugation. That's 2. And then transformation. Is that the 3rd one? Correct me if I got that wrong. I'll take a look here. That's the operon. You should know about mutations. Oh, there it is. I got them right. Transformation, conjugation, transduction. All right, sorry about that. My mind was freezing here. I don't have this lesson prepared. I'm just trying to give it to you. Let's see. You should know how replication differs from transcription and how transcription differs from translation. All right, any question about chapter 8? All right, those are all the chapters that we had quizzes on. And so, with the exception of maybe chapter 1 and chapter 8, there will be at most 2 questions from the chapter. Chapter 1, because it concerns history, there will be a few more questions. And then chapter 8, because we spent so much time on it. If you look at the syllabus, I think we spent close to 3 days on that chapter. There will be a few more questions on chapter 8 than the other chapters. Not too many more. Now chapter 7, chapter 13, and chapter 14 will have a large number of questions on them because you haven't been quizzed on these chapters yet. Let's take a look at chapter 7. You should know the terms, be able to discuss the factors that influence microbial death rates, like greater time is needed to ensure that all the microbes die, so greater time of exposure to a disinfection. If there's more bacteria there, then they're harder to kill, so you need even further time. If there's organic matter around, it could inactivate the disinfectant, etc. Be able to discuss the three main ways in which antimicrobial agents kill or inhibit cells. Can anyone mention the three ways? Anyone want to guess on one of the ways? Inhibit? Come on.
Breaking the envelope?
Yeah, inhibit the microbial envelopes like the cell membrane, or if it's a eukaryotic cell, the nuclear membrane. So that's one way. Damage DNA and denature proteins, especially enzymes. Those are the three main ways that antimicrobial agents kill or inhibit cells. Be able to understand the various types of disinfectants and how they work. I went through that very quickly in the table here. And remind me at the end or in the lab, and I'll try and narrow down the question on this. I remember I'm asking, I'll tell you, I'm going to ask you about halogens, okay? So you should know this table for halogens, okay? I remember that one. I don't remember if I have any other questions. And I feel you should know halogens because you've had exposure to bleaches and bleach and tincture of iodine. I think everybody's had exposure to those, and those are halogens. Okay, and then you should understand the relative resistance of the major microbial groups to the antimicrobial agents. And I tried to go over that here, where we're looking at the resistance of the microbial group to the agent, and these are the most resistant generally to the least resistant. But I did discuss that there are exceptions. Where do I have that down? Oh, here it is. Mycobacteria are the third most resistant, but yet, because of their cell wall, they are very sensitive to alcohol, which is not terribly a good disinfectant agent. In fact, it's a lousy disinfectant agent. We don't even call it a disinfectant. We call it a degerming agent. But mycobacteria are very sensitive to them. And so that's an exception. And then you should know endospores are very resistant to most disinfectant agents. And that's because that endospore structure resists almost everything, even radiation. And there's only a few antimicrobial agents that kill endospores. And the common one is hydrogen peroxide and other peroxides can kill endospores. All right. Is there anything else I want to talk about here? I think that's it. Know the terms. Any questions about Chapter 7? Chapter 13. I might be able to finish this during the lecture. Oh, Chapter 13 on viruses, viroids, and prions. We should know all three of these, what they are, how they infect their cells. You should know the terms, what's a capsid, what's an envelope. You should know the different life cycles that viruses have, like lytic viruses, and compare them to lysogenic viruses and bacteriophage. And with animal viruses, there are lytic viruses, and there are latent viruses and budding viruses. I don't have that up here, but there are budding viruses. So be able to compare the life cycle of the viruses. You should know the terms, prophage, provirus. This is where the bacteria viral DNA integrates into the host DNA cell with bacteria. A provirus is what we call it when the DNA of a animal virus integrates into our DNA. And that's true for an RNA virus that's a retrovirus, meaning that is an RNA virus, but it goes into a DNA intermediate, and the retrovirus integrates into our host cell's DNA, and that we call a provirus. You should know what viroids and viroids, prions and viroids are. I mentioned that. Understand the characteristics, the structure, and the classification of viruses. Remember with the classification that the simple English word is used for classifying the virus. Like we don't call it a scientific name using Latin, we call it the English name like herpes family of viruses, the herpes family. And most viruses are only classified into families, and then down below that, meaning that you can have genera, species, and then it's important to have the strain, because the strain of viruses can be very different. And I gave the example that with polio, one strain of the virus causes the disease polio, and the other strain of the virus we use as a vaccine to prevent polio. All right? You should understand about viral multiplication, and I've already mentioned about prions and viroids. So know that a virus, its goal is to replicate, and how it does it, it invades a cell and hijacks it, and then uses that cell to make more copies of the virus. Viruses have little or no metabolic activity outside of their host cell. So they only have metabolic activity when they've invaded their host cell. The host range is the range of species that the virus can infect, and that's determined by the attachment factors and then the host cell receptors. Most viruses really only infect the cells of one species. There are some exceptions, like the flu can hit a number of warm-blooded animals, like humans and horses and pigs, but they can also hit birds, which are also warm-blooded animals. You should know the sizes of viruses, viral structure. We already talked about that, and that, and that, and that. Oh, you should know something about coronaviruses and retroviruses, because they're a little bit different viruses. Of course, coronaviruses you should know because of the current pandemic. What's specifically different about retroviruses is they have a unique enzyme reverse transcriptase, which this enzyme can take RNA and make DNA. And this is one case where information does not flow from DNA to RNA to protein. In this case, information is flowing from RNA to DNA, and then the DNA to RNA to protein. You should know how an acute infection is different from a persistent infection, and how a latent infection is different from an acute infection and a persistent infection. You should know about prions. We already talked about that, and plant viruses and viroids. But no question about the huge viruses. All right, any questions about Chapter 13? Chapter 14. Now remember on this one, pay particular attention to the terms. If you know the terms to this chapter, you will know about 50% of the chapter, because terms are really important in this chapter. So know the terms. I've only got a few here. Take a look at the objectives to Chapter 14. You can find them in the notes, and it'll list all the objectives. Know those terms and the objectives. Understand the relationship of normal microbiota and its host. We'll talk more about that in a little bit. Understand the stages of disease development, and that all diseases tend to go through the same stages. Tend to. They don't always. Like sometimes when you get the flu, the prodromal period is missing. Understand what nosocomial infections are and how to control them. And there will be a question on this, because this is something that you should know. Nosocomial infections are also called health-acquired infections. You're going to be working in the health agencies, most of you. That's why you're in this class. So you should know what nosocomial infections are and how to prevent them. All right, any question about any of that? If not, let's go into a little more in depth. Like I said, you need to know all the terms to this chapter just because they're so important in this chapter. You should know that few microbes are pathogens, and some of them are very helpful and you actually require them. And those we call the normal microbiota, microbes that essentially permanently colonize you, meaning their host, or your host, whatever you want to call it. They're only found in certain locations in the body. We got that in the next slide. The skin, the mucosa, which is open to the outside of the body. And the normal microbiota is not usually found in the internal tissues and organs, like the heart. If it is found there, that's an infection. It's not the normal microbiota. Let's see, what was I going to say here? Oh, a disruption of the normal microbiota and the host tends to cause disease because there's a disruption. The normal microbiota is important. It gives us certain nutrients and helps us digest our food. It, on the skin, makes the skin more acidic, which helps prevent pathogens from getting established. Okay? Now the skin isn't too acidic, but most pathogens don't like the acidity of it. A better example would be the urethra. The normal microbiota makes it acidic, and that keeps some pathogens out. Anyways, let's move on. That the normal microbiota differ from the transient microbiota, in that a transient microbiota is only present for a short time, days, weeks, months, but it's not essentially forever, or close to forever, which is the normal microbiota. I don't think I asked any questions about, there's more normal bacterial microbiota in your body than you have normal human cells. And I don't think I asked any questions about the surprising places we found the normal microbiota. But the normal microbiota is important for the reasons I mentioned, to keep away pathogens. They can antagonize the pathogen, compete with them, occupy the space the pathogen needs, competing for resources. If no resources, the pathogen can't grow, producing acids so the pathogen can't grow. And then some of our normal microbiota actually produce bacteriocins, which are proteins that prevent potential pathogens from growing. And bacteriocins are very similar to antibiotics, except they are much more narrow in their spectrum of action. And bacteriocins prevent the growth of only similar bacteria. So a bacteriocin produced by E. coli would only prevent the growth of a similar cell such as Salmonella and Shigella. You should know about symbiosis and the three different types of symbiosis. The normal microbiota is either taking a commensalistic relationship or a mutualistic relationship. And a human pathogen takes a parasitic relationship. An opportunistic microorganism is one that can invade, not normally invade, but after a primary infection weakens the patient, like damaging the mucosa of the respiratory system, so then you're more likely to get a secondary infection. So that's an opportunistic microorganism. And the normal microbiota can be opportunistic. This is especially a problem in people who have compromised immune systems, or patients who have disruption of their skin, like a burn patient or a traumatized patient. You should know about Koch's postulates. That's all I'm going to state. We ran into it 3 times in the class, so you should know about that. I will be asking a question on it. These are more terms. Oh well. You should know the difference between sporadic disease, endemic disease, epidemic disease, pandemic disease. Coronavirus is currently a pandemic in the world, but it started as an epidemic. It was only found in China, but it unfortunately spread. You should know about the severity of disease. Don't remember if I asked something about, well, this is severity of disease, so I already mentioned that. You should know about the patterns of disease, predisposing factors. Now here, let's talk a little bit about the development of disease. Most diseases go through 5 periods of disease: the incubation period, the prodromal period, the period of illness, the period of decline, and the period of convalescence. You should know what each of these periods are and what the disease is doing in each of these periods. Now, with that stated, there are some diseases which don't have a period. Like I said, the flu sometimes does not ever have a prodromal period. You're in the incubation period, you don't know you're infected, and then all of a sudden you're in the period of illness. It's like somebody hit you over the head with a hammer, sledgehammer, I should say. And then some diseases do not have a period of decline or a period of convalescence. And those are the diseases where the disease usually kills the patient. And HIV and AIDS is an example where the patient never enters a period of decline or a period of convalescence. You should know about the spread of disease, spread of infection, transmission of disease. I will not ask you about the difference between an airborne transmission and a droplet transmission, because I think it's arbitrary, the distinction that this is droplet transmission and then after this point here it's airborne, and I just think that's a little bit of an arbitrary distinction. I won't ask you about that. Vector transmission, and then know about nosocomial infection agents, okay? It's important because you're going to go into the healthcare facilities. You should know a little bit about epidemiology. All right, any questions about this lesson? I have not reviewed about the lab. Are there any questions about the lab? Remember, about 10% of the questions in the final will cover the lab, so you do need to study the lab as well. And I've highlighted the material you need to know in the lab.
Oh, thank you.
All right. All right. If there's any questions in the lab, I'll be there from 6:30 to 6:45. All right? And I'll see you whenever. All right.
All right, thank you.
All right.